Active transport is the transport of molecules against its concentration gradient, which means that it would be going from a place of lower concentration and pushing through to higher concentration. It is going against what it would naturally do. It's going against its concentration gradient, and so it requires ATP. This is considered primary active transport. We'll talk about what secondary active transport is in just a second. So as you can see in this picture, this is considered primary active transport because it is just taking a calcium ion from an area of low concentration to an area of higher concentration, requiring ATP. Secondary active transport is going to use the energy that was set up by that primary active transport to drive something else along with it. So this is kind of bringing something along. So it's also called coupled transport. This is considered active transport because ATP is required for that initial concentration gradient in the first place, and this usually deals with sodium. So we have a couple examples here for you. In this bottom picture, it doesn't show the ATP being used and the sodium concentration way higher on the outside than inside the cell. But what is known here is that the sodium potassium pump has used ATP to push sodium out of the cell against its concentration gradient. And then other protein channels are going to use that energy from that concentration gradient and push along this glucose molecule with it. And so sodium is wanting to move along its concentration gradient. Glucose is moving against its concentration gradient here, but it's coupled with the sodium ion using that energy. And again, this is why it's called secondary active transport. This is called co-transport or symport because they're moving both in the same direction. They're going in the same direction. And then there is the example of counter-transport or antiport. And that example that I just mentioned was our sodium potassium pump. In this example, you can see that ATP is required. Potassium ions are being pushed against their concentration gradient so that there is going to be a big concentration of potassium inside the cell, a really small one outside the cell, and then the opposite is true of sodium. Sodium is also moving against its concentration gradient, but it's going to be highly concentrated outside of the cell, less concentrated inside the cell. This requires ATP because it's going against their concentration gradients. This is called countertransport or antiport because these two molecules are going in the opposite direction, unlike symport in the last slide. In anatomy, we spoke a lot about how absorption occurs, and this is a great image of showing you how the sodium potassium pump and co-transport allows us to take in glucose from our small intestine. So here we have our sodium potassium pump making it so that the concentration of sodium is really low in the cell, but greater elsewhere, right? And maybe it's greater out here, and maybe it's greater out here. And we have that co-transport example where glucose is going from a lower concentration, so it's going against its concentration gradient here, but it's like hitchhiking on the back of our sodium, which is going to rush in because of this already established concentration gradient from the sodium potassium pump. This is how we absorb glucose into our epithelial cells in our intestine. And then notice glucose concentration is higher in the cell, and now to get into the bloodstream, it goes just via facilitated diffusion through a protein channel. This is how we absorb glucose through our epithelium. It is active because it is requiring that concentration gradient that is set up by the sodium-potassium pump in the first place. Some other examples of active transport are bulk transport. Bulk transport means they're going to carry something large, and there's a few different mechanisms. They're called phagocytosis, which has been discussed in anatomy and elsewhere, endocytosis, and exocytosis. Phagocytosis. You've probably heard that our white blood cells do phagocytosis. I would always say like they kind of eat up these foreign invaders or particulate matter into the body. And so this is showing you a white blood cell here. And what they do is they form these pseudopods. They're called pods because they kind of look like legs. And it's kind of invaginating around this particle to intake it in, like forming a vacuole or a vesicle around it. Our white blood cells do this, our macrophages do this. And then there's different types of bulk transport where we take something in, and that is called endocytosis. There's two main types of endocytosis. First is penocytosis. I think of peno, and I think of this as like, I think it literally means to drink, kind of like Pinot Noir or Pinot Grigio. And that's because it's kind of when our cell invaginates inward and takes a drink of the extracellular fluid. If it's taking a drink of the extracellular fluid, that means that it is not necessarily looking for something specific. It is non-specific. It's just taking in some of that extracellular environment. This is different from receptor-mediated endocytosis. Receptor-mediated endocytosis have receptors on the surface of the cell, and they are looking for particular molecules that are going to bind to it. And when those particular molecules bind, it will cause that invagination, and we will have the vesicle with those specific molecules that were bound to those receptors. So different from the penocytosis, this is specific. It's looking for some specific molecules because it's receptor mediated. Last, we have the opposite of all that, and that's called exocytosis. Exo means out, endo means in. And so this is talking about removal or, dispersal of of molecules from the cell. So we have a vesicle here. It's going to fuse with the cell membrane and secrete its products. It's literally the opposite of endocytosis. Endocytosis is shown in this first picture. And if you didn't know this from Gen Bio or from Organelle's review, vesicles are made out of the same phospholipid bilayer as the cell membrane is. And so when we have endocytosis and endocytosis and exocytosis, they literally just fuse with the cell membrane and kind of pinch off or, join in for allowing these things in and out. So a quick review of neurophysiology or of chemistry. Like charges repel. So if you've ever held two magnets before that are, pluses are going to repel each other. Negative ions are going to repel each other, whereas positive and negative are going to attract each other. Cations is another name for positive ions. Anions are another name for negative ions. And then what we're going to talk about right now is called the membrane potential. And what that is the separation of opposite charges across the membrane. And so we mentioned before how we have a lot of varying concentrations of ions outside the cell and inside the cell, and that is going to impact its membrane potential. We can calculate a membrane potential using a voltmeter, which I'll show you in just a second. But here's that image. Here is that image from before where we have varying concentrations of ions due to our active transport. That's going to change that, but that is going to have an effect on what the membrane potential is. So we have all of those ions, but we also have a lot of ions or anions. They have negative charges that cannot move out of the cell. They have to reside in the cell. These are large proteins, organic molecules, phosphates that are too large to move through the cell. That is going to make positive ions, cations, attracted to coming inside. So they're going to want to come inside. So that means, you know, at any given time, cations would like to come into the cell. But the concentration gradient is going to be high if this is going to occur. The concentration gradient of cations is going to be high, and the concentration gradient is going to push these cations out from higher to lower concentration gradient. So there's two mechanisms that are going to impact where these molecules are going to lie. The electrical attraction, you know, which these anions are going to impact, and then the concentration gradient. So there's two separate things that are going to impact this. So let's say for a moment if our cell was only permeable to potassium. We have these fixed ions here, these fixed anions here, which is going to make potassium really attracted to coming inside. But then the concentration of potassium is going to be really high inside the cell, and so it's going to drive the chemical gradient will drive potassium ions out. This will happen until potassium ions reach an equilibrium. And when these potassium ions reach an equilibrium, the electrical gradient here that you can measure with a voltmeter is about negative 90 millivolts. And so what that is that's considered potassium's equilibrium potential. Meaning potassium will reach an equilibrium where the electrical attraction and the chemical attraction are going to reach an equilibrium, and this will be the voltage because of those anions. So this is kind of just like to show you what it would be like, but really what we have is, in addition to the movement of potassium, and potassium does actually leak through our cell pretty easily, we also have some movement of sodium in. And so our resting membrane potential for any given cell is somewhere around negative 70 millivolts. So the resting membrane potential is the membrane voltage of the cell when the cell is at rest. It is not producing any impulses. So it's around negative 70. If we want to include like most types of cells, it can range anywhere from negative 65 to negative 85. And they have a negative internal charge mostly because of this large anions being stuck inside the cell, also because of the sodium-potassium pump. And sodium actually cannot move through the cell very easily. As I said before, potassium can actually leak pretty easily, and remember that its resting membrane equilibrium potential is negative 90 millivolt. So this is just slightly more positive than that. A good way to remember where sodium potassium ions lie, I would always tell my students that our cells are like bananas in the ocean, because bananas, as we have been marketed, are full of potassium, and the ocean is full of sodium. And so what that means is that our cells have a higher potassium inside the cell in the intracellular fluid. I said the wrong thing. Potassium has a higher concentration inside the cell in the intracellular fluid, less so in the extracellular fluid, more sodium in the extracellular fluid, less sodium inside the cell. So back to that sodium potassium pump. It uses ATP, moves three sodium out for every 2 potassium in, and they are moving against their concentration gradients. There is going to be slight leakage of our potassium. We'll talk more about that later. And again, this is an example of antiport. This is really important. Our concentration gradients that are set up by the potassium pump is actually what allows us to generate our nerve and muscle impulses. It's literally what allows us to communicate, it allows for communication in the body. So this slide is showing you the different types of chemical signaling between cells. Thanks to our sodium potassium pump, these types of signaling are possible. The first one is called paracrine signaling, and I like to think of this as a localized effect where you have a cell that's secreting the signal and a local cell that's receiving it. That's a paracrine regulator. One that is not shown is called autocrine. Auto means self, right? So say these cells that secreted the signal also received it, and that would be considered an autocrine signaling. Here we have synapses, which is what we're going to get into next, about how our nervous system communicates with the rest of the body. And then down below we have our endocrine signaling, which is our hormones being released by our endocrine glands, going into the bloodstream to reach the target organs that have receptors for those hormones. Depending on the polarity of a molecule, a regulatory molecule, those signal molecules we were just talking about, the receptors for these molecules are going to look a little different. If the regulatory molecule is polar, meaning if it's water soluble, we will have receptors on the surface of the cell. Notice that they don't have to go inside the cell on their own. They're going to bind, and then it causes kind of a cascade effect where it impacts secondary messengers and it's going to change the function of the cell. If a regulator is nonpolar, meaning if it's lipid soluble, like a fat, like a steroid, for example, it can actually go through the cell membrane and its receptors will be in the cell, so intracellular. It's going to bind, and then that will cause a change, and notice it's impacting the M and mRNA, which can produce a different protein here. G protein signaling is a different type of signaling where they have 3 proteins attached to it. 3 proteins attached to a receptor, and these 3 proteins can actually disassociate. They can come off, and we'll talk about what they do. They are called alpha, beta, and gamma. Alpha, beta, gamma. And so a regulatory molecule or a signaling molecule is going to bind to the receptor. The alpha protein is going to release GDP and take in GTP, and that is going to cause either the alpha molecule or the beta gamma molecules to be released and to attach to an effector. And so you know what an effector is. An effector is going to turn on and cause a change, cause an impact, right? Have an effect. And in that way, this is a different type of chemical signaling.